The protocol combines the power of generating human cardiovascular cell types using induced pluripotent stem cells and the versatile technique to generate beating cardiac spheroids in less than 72 hours. These cardiac spheroids can be used for disease modeling and drug testing. One of the main advantages of this facile technique is that one could generate nearly 1,000 beating cardiac spheroids in a standard 12-well plate. And most importantly, each and every cardiac spheroid can be assayed for its contractile function using imaging-based technique.
After melting agarose in the microwave, place it in the biosafety cabinet and allow it to cool for 3 minutes. Pipette 700 microliters of the molten agarose and add it to the silicone micromold of a 9x9 array, taking care to avoid generating bubbles. Carefully place the mold on the pre-cooled ice block to accelerate the agarose gelation. Once the agarose becomes translucent, carefully bend the edges of the micromold to loosen the agarose replica and gently peel the replica from all sides to detach it from the silicone micromold. Transfer the agarose microtissue tray containing 81 circular recesses into a sterile 12-well plate. Then add 2 milliliters of PBS to the agarose microtissue tray and inspect it under the microscope for any trapped bubbles or irregularly shaped wells. Submerge the agarose tray in 2 milliliters of 70% ethanol.